Hello, good morning everyone. Today we are going to learn about Meselson and Stahl experiment. Francis Crick said that DNA replication is semi-conservative in nature, but there was no conclusive evidence to say that DNA replication is semi-conservative. Meselson and Stahl they did experiment which proved that DNA replication was semi-conservative. Let us know what was their experiment, how did they do their experiment. Meselson and Stahl, for their experiment, they used nitrogen isotope. It is isotope, not radioactive. What is meant by nitrogen isotope? They took a nitrogen isotope of N15 and N14, which means if you take a normal nitrogen, which is N14, N14 is the mass number, or you can consider it as atomic weight. What is that mass number? 14 refers to in a nucleus there are 7 protons and 7 neutrons together constitute 14. That is normal one. They took heavy isotope of nitrogen. Heavy is N15. What is N15? 7 proton, but N15 has one additional neutron, hence 7 plus 8 in the nucleus will make it 15. 15 compared to 14 is heavy, hence N15 heavy isotope of nitrogen, N14 light isotope of nitrogen was taken for the experiment, which means in this N15 and N14 they were taken in the form of ammonium chloride. Uh, if you take ammonium chloride and if you grow the cell containing DNA, the N15 or N14 gets accumulated in the DNA. Where exactly in the DNA N15 and 14 gets accumulated? You know that DNA is nothing but nitrogenous base, sugar and phosphate. Nitrogenous base, there is one nitrogenous base called thiamine. In the thiamine, in its first position and third position, there is nitrogen. There N15 or N14 can go and accumulate while forming DNA. So DNA, in the DNA, N15 and N14 gets accumulated in their thiamine bases. That is first thing they took for their experiment, nitrogen isotope. Second thing they took was cesium chloride salt solution. Why cesium chloride? During the experiment, they did ultracentrifugation, giving more rotations to a solution. Their centrifugation was based on density gradient. Gradient refers to where one side there will be more, another side there will be less. That is the meaning of gradient. So they wanted to separate the substances present in the centrifugation tube based on the density, which means heavier will be settling down, lighter will be at the top. To separate them, they took cesium chloride salt. Why cesium chloride is? Because density of cesium chloride is 1.71 grams per centimeter cube, which is equivalent to density of DNA. So you may ask, what is the density of DNA? It is same, 1.71 grams per centimeter cube. So that is the cesium chloride salt whose density was equal to DNA was taken for density gradient centrifugation. That was the second thing they took. Now let us begin with their experiment. First they took N15 isotope solution. In that, E. coli was grown for many generations, about 14 generations they grew. E. coli, its doubling time is 20 minutes. For every 20 minutes, cell will double. One cell will become two. So every generation they observed what was the DNA, what isotope was there in the DNA. Now they have taken only N15 isotope. So E. coli cells which are growing will be having only N15. There is no chance of accumulating other isotopes. So they observed that particular sample. If you are growing the E. coli cell in N15 and if you are observing, that is called zeroth time. In the zeroth time, by doing cesium chloride density gradient centrifugation, they observed this is the centrifugation tube. Here lower region which I have marked corresponds to heavy band, heavy region. Middle one is medium and the upper one is lighter based on the density. Their centrifugation tube showed a band, a dark, sort of thick band at the region corresponding to heavy, indicating that all the DNA present in the E. coli cell, they just had heavy isotope N15. So 100% heavy, which means DNA, both strands were labeled with N15. N15 gets accumulated in the thymine base. In the next part of their experiment, few cells of E. coli from N15 isotope was taken and transferred into the medium containing N14 isotope. N14 isotope is in the form of ammonium chloride. What is done? Cells of N15 was taken and transferred to N14. Now they observed for every 20 minutes, because doubling time is 20 minutes, as I said. For the first 20 minutes, which is regarded as first generation, they centrifuged using cesium chloride. They got a band which was neither in the heavier region nor in the lighter region, but it was in the medium region, indicating that the DNA is neither heavy DNA nor lighter DNA. Instead, it is a hybrid of two. So they analyzed that data. Why that hybrid band was got? Because they found out that one strand was N15, one strand was N14. That's how you can get hybrid band, no other ways. One strand was N15, another strand was N14. After 20 minutes, one cell will divide into two, so two DNA strands, hence indicating after 20 minutes semi-conservative replication takes place, which means they use these N15 as template strand, and a new strand which is nothing but having N14 is synthesized. After again 20 minutes, that is 40th minute, second generation was observed. Centrifugation was done. They got band corresponding to medium region and a light region. Bands were of same thickness, hence 50% hybrid, 50% light. Two cell will become four DNAs have been labeled. When they analyzed data, they found two hybrid, two light, because of semi-conservative replication. Further, 60th minute, third generation was observed. Third generation showed that there was a thick band corresponding to lighter region and a thin band corresponding to the medium region, indicating 75% of them were light, 25% of them were hybrid. 4 cell has become 8 now. They use all these as template. 1 template, 2, 3, 4, 5, 6, 7, 8, using all these 8 template, and the lighter DNA, new DNA is synthesized. Therefore, you are going to get 2 hybrids and the remaining 6 will be lighter strand. After 80th minute, 4th generation was observed. There also they observed that there was semi-conservative replication. They got 88% light band and a 12% hybrid band, indicating that when they analyzed data, it showed that previous strands were used as template strand and a new strands were synthesized using N14, hence 88% light, 12% hybrid. So Meselson and Stahl, finally they came into conclusion that DNA replication is semi-conservative in nature. Thank you. If you have liked it, please click the like button.